In this video, we will assess a PAH missense variant for eligibility towards ASO therapies. Step 0 is a variant check, which requires us to check whether the variant is correct. In this example, the variant is correct. We next check the inheritance pattern. As recommended by our guidelines, we can use OMIM to check for this. As we can see here, the phenotypes are a result of autosomal recessive inheritance. Next, we check the variant mechanism. If we go back to OMIM, we could see here that most missense variants found in the PAH gene result in misfolding, increased protein turnover, and loss of enzymatic function, and that decrease in protein stability is the main pathogenic mechanism. If we conduct a literature search for our variant, we can see that our variant results in loss of function. For instance, this paper here quantified the residual activity of multiple variants, including our variant of interest in a compound heterozygous patient. No residual activity was detected for this allele. For those interested in learning more about these variants and the methods used in quantifying residual activity, the PubMed ID can be found on the bottom left. Now that we know our variant results in a loss of function, we next check whether this variant affects mRNA splicing. If we look up our variant on ClinVar, one of the databases shared in our guidelines, we can see that this specific variant has multiple entries. The first entry here, from an FDA-recognized database, makes note of alternate splicing, resulting in a 96 nucleotide deletion, and includes a PubMed ID link to the corresponding study. The PubMed ID links to this paper by Ellingsen et al. In this paper, they note that the mutation creates a fully active, novel 5' donor splice site, which results in a 96 nucleotide deletion at the 3' end of exon 6. Now that we know this variant affects splicing, we can proceed with evaluating the variant for eligibility towards splice-correcting ASOs. Since this is an exotic variant, we need to check the distance of the variant from the canonical splice site and the effects of the amino acid change on the protein. In the same paper by Ellingsen, they noted that the missense mutation itself resulted in a small reduction of protein stability and enzyme activity, and the loss of function effect is mainly a result of the alternate splicing. Using the UCSC genome browser, we could see that the location of the variant is in the center of the exon, well past the 50 nucleotide threshold, posing no concern for effects on the canonical splice site. From our search, we know that there are functional studies which validate the effects of alternate splicing, and that the canonical splice site is not destroyed. Additionally, the donor splice site is not within 15 base pairs of the canonical splice site. However, there is a small reduction in protein stability and enzymatic activity due to the amino acid change itself. Therefore, we cannot classify this variant as likely eligible. If we scroll down, we see here that variants that produce a partially functional protein are classified as unlikely eligible. We cannot say for certain whether or not the residual activity can ameliorate patient phenotypes. In real-world scenarios, when working with real patient data, taking into consideration other factors, such as whether the patient is compound heterozygous or homozygous, are all important factors that must be considered. For now, we classify this variant as unlikely eligible.